All right, so now we've seen these first three steps in what's going on with your lymphocytes that started out in your bone marrow and then continue to mature on and become those adult lymphocytes, if you will, and develop those B cell and T cell receptors in the bone marrow or in your thymus. Those cells have receptors that have a variable region that can bind to foreign objects that are inside of your body, and they head out into circulation and again go throughout your bloodstream and hang out in various elements of your lymphatic system, which if you've taken anatomy, you know more than you ever wanted to know. But remember, you've got lymph nodes scattered throughout your body that are filtering fluids using these lymphocytes to grab on to the foreign objects that are out there. That's why they're hanging out in your circulatory system and your lymphatic system. And remember, your lymphatic system also includes things like your spleen that is constantly filtering your blood as it courses through your body here. So if we look at these lymphocyte responses, and we'll take a look in step 4 here, and we'll start out looking at our T cells in what's known as cell-mediated immunity. And this is going to do two things really. They're going to act on foreign objects that are inside of cells or cancerous cells, and they're also going to boost the immune system. And this leads to the question, well, how can a T cell attack an infected cell? They need to see what's inside of that cell. And that's where we see something that's really kind of interesting that is known as the ability of these cells to display particular epitopes from the inside. So they're going to take foreign molecules and cut off chunks of them and then stick them onto something that is known as MHC for major histocompatibility complex. And this was first identified when they looked at transplant patients that were rejecting various organs. They found it was because they had a different MHC on the donor versus the recipient who got the organ there. And so the recipient's immune system attacked these tissues because it was able to recognize that the MHC on the surface of the organ was actually different. And you can see here, we find MHC class 1 over here is on the surface of all cells that have a nucleus. So you're not going to find this on the surface of something like a red blood cell that's out there. And this is basically going to present things from the inside. So it's known as cytosolic or endogenous pathway, because you're going to take self-proteins and also cells that are infected with viruses that are making viral proteins. Those are actually going to get chopped up and loaded up onto this MHC class 1. MHC class 2 over here is what's known as the extracellular protein pathway. And this is where cells in your body are going to be taking particles that are outside and bringing them in. So think macrophages, and I'll show you a little cartoon to show you how this process actually works. So if we look at this little cartoon, we can see two different cells that have foreign objects. In this case, you've got a cell that's infected with a virus, and that virus is going to hijack the ribosomes. And so as those ribosomes are making viral proteins, just every once in a while, we will actually degrade some proteins and take little chunks of those and display them. If it's self or you, your body's not going to recognize that thing as different. But if you've got an infected cell that is actually making viral proteins, now you're going to display chunks of those viral proteins on the surface of that cell, and that cell is displaying something that is foreign as a signal to your immune system to attack it. And over here we see what's known as an antigen presenting cell. This could be a phagocytic cell that is doing endocytosis there. So remember, chemotaxis, it moves towards the foreign particle, it ingests it, and then you get your phagosome that fuses with the phagolysosome. Well, you break down this antigen, and then your body is going to take little epitopes from that antigen and load it up to MHC that is inside of a little vesicle right there. And then this vesicle can go to the surface of this cell, and now that vesicle is displaying chunks or epitopes from the pathogen that was actually ingested. So you can see two different ways of presenting things from inside of the cells there. So if you look at this, you can see that this little guy over here is going to be presenting things from the inside, like our virus-infected cell, and this one over here is going to be presenting things that were scavenged. And you can think of it as it's part of that whole system of your immune defenses actually boosting things. In this case, with MHCO2 over here, we can take foreign particles, eat them, and also use the leftover bits to sort of wake up your immune system by presenting some of those epitopes from that antigen in this MHC molecule right there. If we continue on and look at this process here, that T cell that is out there is floating around, and it's going to actually have the ability of getting activated by binding to that MHC. And I'll show you in a figure in a second here, when we look at this process here, this activated T cell is going to be able to divide. And this is sort of a general idea here is the T cell is going to get activated and it will divide like we saw earlier. And when it divides, it's going to be doing mitosis. So it's making clones of the original T cell. And we're going to get two different populations of cells out of here as they differentiate. You're going to get your memory T cells over here that have the exact same T cell receptor on the surface right there. And now you went from one T cell to potentially hundreds of T cells that are being made right there. The other thing that's coming out of this is you've got your effector cells. These ones are going to be leading the attack on the cells depending on if they're infected, or you will also have some T cells that can boost the immune system here. So when we look at this process here, it's important to recognize that when these lymphocytes, B and T cells, are activated, it's going to cause them to divide, and you're going to start getting pools of cells that are out there. The memory cells are saved for later, and they can distribute throughout your body so that when that antigen comes back, you'll be ready to mount that immune response quicker and faster. And if we look at this process, we can look at the effector T cells that come out of this. So your memory cells are hanging out ready for the next time, but you also want to have effector cells that are actually going to work immediately to start mounting those immune responses. One of them that comes out of here is what's known as a regulatory or a suppressor T cell. And these guys are basically out there to prevent excessive responses. They're also going to be out there if you develop an autoimmune disease. The regulatory T cells can start to hopefully dampen that immune response. And again, this is where we see the problems with the non-specific defenses. If you have a chronic infection going on, you're going to keep causing damage over and over to the innocent bystanders. And so those regulatory T cells are going to help to at least lower that immune response so you don't cause too much damage to yourself there. If we look at the second pool that can come out of here, are cytotoxic T cells. These are the ones that are going to destroy the target cells that are infected or cancerous. And then the last one that comes out of this process are the helper T cells. And these are the ones that are going to help to boost your immune system, your lymphocytes, including B and T cells, but also all those other kinds of white blood cells that we saw out there. And so we'll take a look at this process and see how they're going to play a role here. And if we look at this process, we'll start out with cytotoxic T lymphocytes. These guys are the ones that are going to be responding to the intracellular pathogens or things like viruses and also cancerous cells that are out there. And so if we take a look at this little example over here, you've got your infected cell right here. It's got a pathogen on the inside, and we are displaying a little epitope of that pathogen right here that went through this cytosolic pathway right there. So here we are displaying it on MHC1. So if you have a virus on the inside, you chopped it up into little pieces and you stuck an epitope onto MHC1 that went out to the surface of the cell right there. Well, along comes a naive cytotoxic T cell right here, and on the surface of this cell there's actually two different receptors that are going to play a role here. One of them is our T cell receptor, which you remember, it has a variable region. And that variable region can bind to our epitope that is displayed on MHC. So we can see it blown up right there. Your T cell receptor has the variable region with the hand that can grab on to the epitope. And if you think about it, this could happen sort of randomly with things that are floating around. And so what your T cells do is they actually have a double handshake. They've got a T cell receptor and then what's known as a co-receptor. And in this case, it's called CD8. And that co-receptor is almost going to reach out and check to verify that your MHC1 is actually present there. So that's going to prevent this from just having an epitope bind and randomly triggering this. You actually have to have the MHC and the epitope present to actually activate this T cell right here. And once that thing is activated, now you're going to see the cytotoxic effect here. And that T cell is going to start secreting various chemicals into this region right there. So you can almost look at this thing not only triggers the T cell to start shooting, but it also holds it in place so that it doesn't just float off and randomly start to release these cytotoxic chemicals right here. One of them that's released is known as perforin, and that chemical is basically going to perforate, and the other one is granzyme. And I wanted to show you this little guy right here. I got this from a Nature review on immunology, and it shows you these little granules that are being fused, or vesicles that are fused with this membrane right there, and your T cell is going to dump all of these cytotoxic chemicals right there. The perforin is going to start forming pores, kind of like what we saw with complement, but that isn't quite enough. And what the cells do is they also release granzyme, and the granzyme will actually get into the cells and it will trigger apoptosis, that idea of programmed cell death. So if you think about it, this cell over here that was presenting the virus protein, it's now going to commit suicide. So even if the virus is there, its ribosomes for the cell are going to shut down and it won't be able to make more viral particles right there. So really kind of a cool idea that the cells that are displaying the foreign epitopes are basically signaling, hey, I am infected, you need to take me out of the population here. On the other side, you've got these CD8 cytotoxic T cells floating around out there. We've also got a population of T cells that have another molecule on the surface that is known as CD4. So these guys are known as CD4 T cells, and they are going to be the helper T cells, as we'll see. So imagine you've got your antigen presenting cell over here. In this example, it's a dendritic cell that you would find in something like your epidermal layers, and they send out all these little dendrites that are going to serve to increase the surface area of the cell so they can do more phagocytosis. This little guy eats something, and it's going to chop it up and take epitopes and display them on MHC2. And similar to our cytotoxic T cells, along comes a naive helper T cell that has its T cell receptor on the surface, and it also has its CD4, which is analogous to CD8. If we look at the blow up here, we can see that same process that we saw with the cytotoxic T cell, with the key difference that we are presenting this as an exogenous antigen, so it was from the outside and it's displayed on MHC2 over here, and your T cell receptor does the same thing. It binds to the epitope that is displayed on that cell, and here we have CD4 checking so that you don't have to worry about this T cell receptor binding to the wrong kind of MHC that is displaying that epitope there. And if we look at this process here, we can see the stepwise fashion right there. Your antigen presenting cell, or APC, is displaying the antigen. Your T cell receptor binds. And when this happens, that antigen presenting cell is going to secrete one of those cytokines that's out there that I talked about before. And in this case, the APC is going to secrete interleukin-1 or IL-1, and that is actually going to activate that T cell to divide. So the binding not only stimulates the T cell, but also this thing is releasing a chemical to increase. It's almost like this cell is saying, hey, I've got a bunch of good stuff here. You need to make even more cells because we want to help our T cells or our immune system out. And again, we're going to see a production of memory cells and we're going to see the production of our effector cells.